All right, let's introduce your toolbox. Here it is. This is a version of it. I am sure this will change as we go through the term. These are some things that I find useful. So, what's on here? These are a lot of what I would call mid-tier tools. These are things that are available as online web servers, meaning that you, I, or anybody can go there, submit a bit of code, and within a few minutes or sometimes a few seconds, get back a bioinformatic prediction result. So they're like little tools. Each one is good at one, maybe two things, and pretty bad at everything else. They all have certain things they require, or, there are different ways to break each one of these things. The other thing they have in common is that none of these is necessarily the best way to do this. There are standalone tools that will do pretty much all of the functions that you see here, and that at least on some data sets will perform a little bit better than these, but these are pretty good. And these don't require you to install anything on your computer, so that's why I like them for a course like this, where we have a whole bunch of who knows what kind of computers you even have. All right, so let's take a look, and then I'll make a series of videos where we go through each one of these little things and talk about what's there, what's not there, how's it work, what do you do with it? Yeah, this is like a carpentry course, in the sense that we're not working with wood, but we are using tools that will do certain things, and people who know how to use the tools more skillfully will make better products, better bioinformatics predictions. All right. And the other thing these have in common is that most of these can only handle a few sequences at a time. Now, big time, powerful bioinformatics, the things that people pay big money for, these are going to use tools that are doing the same thing as you see here, but they're able to process millions or billions of sequences at the same time. And it's just a more efficient way of finding that needle in the haystack, or assembling that haystack into a little hay house, something like that. It's a nice way to do it. So at the top, we have our sort of main basic tools. So for DNA or RNA searching, stuff like that, GenBank has all the genes. This is a good place to go. But there are many sequences where we read it in little tiny chunks. This is called next generation sequencing. And a lot of these remain unassembled, and you can still search through them. And the cool thing is there are wonderful things in there that nobody has found because nobody has assembled them. So yeah, that's your sequence read archive. Now, if you want to get old school, your sequence is going to come not just as a series of letters, because any fool can read the letters A, C, G, and T. Yeah, no offense to people who have a hard time with that. But what you need to do is assess whether or not you can believe those letters, because even though it looks like the letter is on the page, as with everything, you've got to take it with a grain of salt, and you got to take it with a little probability. So Teal is an online viewer that lets you look at the most common version of old school sequencing. It's called an AB1 file, and these come from older sequencing machines that are still used by labs, including mine, to this day, because they're very convenient and they give you nice long reads. Yeah, and we like that sometimes. Okay. IUPAC uncertainty codes are just ways of dealing with uncertainty. If you have a mix of A, C, G, and T at a particular position, so you know this is the nucleotide, but it can be any one of those, then there's a code for that, which is N. There's a code for anything that's not A, so a mix of C and G and T, and that would be B, which is not A. This is a list of all these codes. They are worth looking at. They are worth keeping in your mind, because you're going to see them occasionally, and you may need to use them occasionally, and it's a good thing to have in there. All right, then we get to some of the search things. So BLAST is a search tool it's like the Google for genes, basically, genetic information, including things that aren't genes, that are kind of in between genes, but on a chromosome, or little bits of DNA that somebody found in a pond. It's all in there. And you can search it very quickly, surprisingly quickly, really nicely. You got the similar things for that are going to start with a protein or end with a protein, and these are all versions of BLAST. HH-Blitz is a different kind of beast, whereas BLAST is searching for little bits of each protein. Like you take a protein that's got lots of little single-letter amino acid codes, and you break it down into, say, okay, that's a word, this is a word, and you compare, like, okay, I found this word next to this word, and you see how many times that happens in the database just by chance. And yeah, if you get a whole bunch of words in a row that all happen to match, then that doesn't happen very often by chance. This is how we calculate probability values. HH-Blitz and down the list HH-Pred. These work on a different thing. So instead of just looking at the words, which they do, they'll also look at conservation in each of the words. So they'll be working from alignments of lots of sequences rather than just trying to align one thing to one thing. And included in that alignment will be information on the structure of the protein that makes, where it's known. And with that kind of information, you can do a much more powerful search, and you can find even very distant things. So HH-Blitz and HH-Pred are awesome at finding distant relatives when you've got a sequence that's just really hard to match otherwise. Other than that, they go in this order. Nucleotide BLAST is good for things that are almost exactly the same and nothing else. Protein BLAST, pretty good for medium conservation things. If you want to find more distant things, you got to go to your BLAST-X or your T-BLAST-N. And those are a little bit more powerful, but they're still using BLAST technology. And so if you really, really want to have a difficult sample, you want some of these HHs, which are using, I believe, hidden Markov models. Yeah. All right, let's keep going. Once you find your sequence, what do you do with this thing? Well, you annotate it. Annotate it just means saying, oh, from here to here, this part is a enzyme. It's a hydrolase, something like that. And based on that, you can say, oh, it probably does this job because it's most similar to enzymes that do this particular job. All of this bioinformatics was really difficult at first, but now that we're starting to get quite a lot of biological information that's actually validated experimentally, we can make much better predictions. So that's how it goes. So let's see. You got all your blasts there. Clustral omega will align any 2 protein sequences or any 5 or 10 protein sequences. It can take a while, though, if you have very long sequences and very many. Once you have your alignment, something like Mr. Bayes is a wonderful way to estimate a phylogenetic tree. So you have an alignment, and the question is, how did we get here? What pattern of mutations and back mutations and sometimes staying the same could get us from A to all the ends of all the branches of a tree? And Bayesian methods are pretty good. There are slightly better methods, but these are pretty close to the best that are out there. And there are different ways of sort of tweaking it. But yeah, this is a nice sort of friendly tool that you can actually use. So we like it. T-Rex is great for looking at that tree, rearranging that tree, setting the root, things like that, once the phylogenetic tree comes out. All right. Annotation. Yeah, so back to annotation. These are, the way we predict all kinds of functions. Like, what does this piece of nucleic acid or protein do? Because that's what we ultimately want to know is, what is this thing? How does it work? So we can find open reading frames that could make proteins. It's not a guarantee that they do, but it's strongly suggestive, particularly if it's a very long open reading frame. Those are very unlikely to happen by chance. XPacy translates a nice little editor that lets you sort of look at a gene, and particularly if there are problems in the gene, it lets you figure out where the problems are and see exactly how we get everything, all the information. So it's nice little nuts and bolts thing. Say you have a piece of DNA. That is actually functionally the same as the reverse complement of that piece. So if we have, let's see, ATG on this strand, the reverse complement of that is going to be CAT on the other strand. And those two are actually the same sequence as far as all of this software goes. Because functionally in nature, DNA and RNA are copied from other DNA and RNA. So if there's one strand, there's also another strand on top of it. It's as simple as that. And so you can always reverse complement a strand and it does not affect, you haven't changed it in any meaningful way. And so if you get a strand and it's backwards, reverse complement it. It'll be forward. You haven't changed it in any meaningful way. Then you can use that and your world is much simpler. So reverse complement will do that with any size sequence. It'll flip it and reverse it. And does that save a lot of time and a lot of manual mistakes. Let's see, HHPred we've already covered. JPred is a structure prediction. So it's predicting secondary structure, which is like alpha helix, beta strand of proteins. Useful. And it's a jury prediction. So it's going to send out your protein to several different bits of software, get all the answers back, and then compare them and say, well, 3 out of 4 pieces of software predicted that you have an alpha helix at this spot with great confidence. And so probably, most likely, that's it. Yeah. It's a nice way of doing secondary structure prediction. Yeah. Not perfect, but just about the best that's out there. You can look for transmembranes, you can look for disordered regions, glycosylation sites, very important on proteins that are going to stick out of a eukaryotic cell, for example. You've got signal peptides for things that are going to be exported to the surface of a eukaryotic cell. And you've got your enzyme pathway database. So the KEGG database is a list not of genes, but of functions that connect to each other. Say you want to make a particular nucleotide. There's going to be a path, and it's going to go, you're going to have several intermediates that lead up to this nucleotide, and there will be a particular enzyme that accomplishes each of those. KEGG gives each of those enzymes involved in a pathway a function, a name that's assigned to a particular function. And so it makes it much easier to understand systems. Like when something changes in the cell and 15 genes are turned on and 15 are turned off, chances are each of those 15 are related genes that are part of a process. Because the cell needs to make something, stop making something, do something, stop doing something. And it usually takes more than one gene to accomplish anything inside of a cell. So yeah, KEGG pathways, very useful for connecting things, connecting some of this data and interpreting it. All right, last real thing here, protein structure. I think this is my favorite part of bioinformatics, and it is imperfect, but it's a lot of fun to actually see what a protein probably looks like in 3 dimensions and rotate it around. Really cool. The protein data bank has all these structures. 3D View is one piece of software that you'll find at the protein data bank that will allow you to show the structures, rotate them around, highlight certain parts of them, stuff like that. It's kind of basic, but it's online and anybody can use it with a computer and probably even a mobile device. So it's good. It'll get us there. But not all structures. So the protein data bank was originally just for crystal structures. But now we have other ways of solving structures. There are NMR structures that usually end up in the protein data bank, so we won't go to their separate database. But there are still some structures that are done by electron microscopy, by just looking at the protein, essentially. And so those appear on the EM data bank, EMDB. There's also an NDB with nucleotide structures, because nucleotides fold up into all kinds of weird shapes, and a lot of those shapes have meaning, purpose, function. Yeah, so that's a nice way. And some of these things are not captured in the protein data bank, so it's a different way to look at this structure. Swiss model will take your protein and do its level best to turn it into a structure by a process called homology modeling. So it'll find things that are similar, or you'll tell it, I know it's similar to this one. and it'll make a structure, full three-dimensional structure prediction for you. Really cool when it works, but it's pretty dependent on whether or not you have a good match to a nice structure. Without that, Swiss model has nothing on which to base its little modeling, and so for completely new and different things, it doesn't have a leg to stand on quite often. Then let's say you actually solve a structure. You've got your X-ray crystallography lab there. How do you figure out, like, okay, it's, that shape. So what? You run it through some of these databases. So Interpro includes the old database that used to be called PFAM, and SCOP is another one. These are ways to look at a structure and then say, oh, yeah, all the proteins that are this particular shape do this particular function. And it can compare them and say, yeah, you're of this particular, we call it a protein fold, which means shape. Unifold will take a piece of RNA or DNA, and it will imagine how it can base pair. How can it make little stem loops? How can it sort of stick to itself? What kinds of crazy shapes will this thing make? Based on a lot of people's very hard work, experimental work in figuring out, like, okay, when I have these sequences, what happens? So it's a nice little predictor. NCBI structure will let you look at these PDB structures, but you can actually, it will mark where alpha helix 1 starts and ends, and it'll show you on the actual sequence. I think that's the one valuable tool you get here. You can find it through Protein Data Bank, but it's really hard to find, and I don't think possible to actually extract in a way that is human readable easily. So yeah, it's NCBI structure is dying, but it still has a place in all of our hearts. And then you can do structure alignment, take the structures of two things and you can align them so that they're both facing the same direction and, more or less oriented the same way. That's useful if you want to show your structure next to the thing that it's like, but you want to pose them the same way so that you can see, oh yeah, okay, mine's a mirror image of the other structure. Last piece of software we'll use, and this is definitely not the end, but it'll be ImageJ, which is a piece of free software that we can actually access through a server. That's the beautiful thing here. And you can do stuff like densitometry, image analysis. You can count how many little green spots there are. Is this cell greener than the other cell? As long as you have the controls there, you are able to actually make conclusions like that. And so it's useful. So you don't just have to, have a picture and point to it and say, there, I think that one's bigger or something. You can measure. Because, frankly, in bioinformatics, nobody can or should ever believe you unless you can attach some kind of error value, like a p-value or an e-value or a z-value to your prediction. Otherwise, you're just saying stuff, and everybody says stuff. We don't have to pay attention to that. We just have to pay attention to stats. All right. Hope this helped, and we will now go through every little one of these tools as a little, I'm going to try to keep it to 2 minutes each explainer. All right, let's go.